Okay, so here we are in a skin cell. This is the nucleus of the cell. And inside the nucleus we have chromatin, DNA. We've got two different chromosomes here. The white string is like one of the chromosomes and the blue is another chromosome. And you can see that over here the DNA is tightly wound around histones. So the part of the DNA that's really tight is not going to be transcribed because it's literally the enzymes that do the transcription can't get, here's RNA polymerase, it can't like fit in because this DNA is too tightly wound. But over here, this DNA is loose. And in the second video, we're going to show you why this DNA is loose and this DNA is tightly wound. The part of the DNA that the RNA polymerase binds to is called the promoter. And the sequence in the promoter tends to have a lot of T's and A's in it. It's sometimes called a ta-ta box. And the T's and A's, when they're hydrogen bonded to each other, they only make 2 hydrogen bonds, where G's and C's make 3. So it's like a little weak place where the RNA polymerase can come and like pull open the DNA more easily so that it can copy one side into RNA. So it's like when you're opening a chip bag and you have that one like weak spot that like opens first. So this is like this weak opening so RNA polymerase can open it. But genes can be turned on and off. And so even though all of your cells have all the same genes, in certain cells, particular genes will be turned on or turned off. So like the metaphor I generally use is like, your genes are like a set of cookbooks, and different cells are going to use different recipes, but they all have the same set of cookbooks. So in the skin cell, we're going to use the recipe for collagen, because skin makes collagen. So this gene 1 is a collagen gene, and so this gene will be on. This gene over here, this is gene 2, we'll say that this codes for a liver enzyme. So this gene will be off in the skin cell, but of course on in the liver. Okay, so this is what happens. RNA polymerase isn't particularly attracted to the promoter here without a bunch of other proteins stuck to the DNA. So this gene won't get transcribed that often, so the gene will be off. In order to turn the gene on, you need something called a hot promoter. So there's a little like weird sexual innuendo that happens mostly because men scientists come up with these terms. So when a gene gets turned on, it gets turned on because the promoter is hot. And the promoter is hot because of transcription factors that are bound to these proximal control elements. So what I've done is I've made the transcription factors look like breasts, because that is going to turn the gene on and make the promoter hot. So the transcription factors bind, they have little DNA binding sites that bind to these proximal control elements, and that makes the promoter hot. And so the RNA polymerase is going to be attracted to the promoter and then copy the gene and then come back and be like, oh my gosh, the promoter's hot, and it's going to copy the gene. So this gene will be hot. And then there's another, if you really want to make a lot of collagen, you need what they call a smoking hot promoter. In order to have a smoking hot promoter, you have to have also activator proteins that bind to the distal control elements, which are also called enhancers. So what I'm going to do with my little model here is we're going to make something that if a sexy woman attracts RNA polymerase, then candy and a sexy woman would make it even more hot. So what we're going to do is we're going to have these activator proteins bind to the enhancer, and that actually causes the DNA to bend. And so now we have this very attractive area. You've got a happy snack and you have a beautiful woman, and so this becomes a smoking hot promoter, and then the RNA polymerase is going to come and bind and copy this gene lots and lots and lots and lots of times. So this gene will be turned on. So meanwhile, this gene is turned off because you don't have any transcription factors or activator proteins bound to these elements. So there's nothing attracting RNA polymerase to these genes. So the liver enzyme gene remains off, whereas the collagen gene is on in the skin cell.